This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 907, recorded on June 7th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi there, Vincent. We've got out my window, clear blue skies, little breeze. Currently, the temperature is 96, headed for 101. And if I look at the forecast for 2 weeks out, all but two of them are in excess of 100 degrees. One this weekend is supposed to be 106.
Wow.
This is a little early for this kind of nonsense, but
It's
that's
hot.
the way it is. That we also need rain. There you go.
Well, right here in New York, it's 24C and drizzling.
No
at least
kidding.
That's what
Wow.
it says. I can't see. And from Madison, New Jersey, Breanne Barker.
Hi, great to be here. It is pretty cloudy outside, and I don't know what the temperature is.
Here it's 24, so it's probably similar.
Yeah, I've got 81 according to my watch.
I was just telling Rich, so we're recording on Tuesday because Friday I'll be at ASM Microbe in Washington, which is a meeting you've been to. In fact, I saw you there.
I have
At
been to
least
that
once.
meeting. Yes, I really enjoy that meeting.
I have a picture one year where you guys all got black lab coats, remember?
We did. Yes, my whole lab had black lab coats.
Yes.
Wow.
I'm doing a TWiV on Sunday with Jeff Taubenberger, who was part of the team that rescued the 1918 influenza virus. So I want to talk about the 1918 pandemic because I have a lot of questions, and I'm sure he can answer them.
I discuss his papers in my classes pretty often. So
Oh, cool. Very good. So I was originally going to do a TWiM on Sunday, which I'm doing. And then Ray Ortega, our producer, said, hey, I have a slot in the afternoon. So I hunted around and I found Jeff, because he's right there at NIH.
Is Ray going to be there?
Yeah, Ray is running what they call, you remember this because you were there for the Kate Rubins episode.
Yep.
It's called the ASM Studio. They have a platform in the middle of the exhibit floor and they do continuous recording. So he's going to be there. Yeah, doing
Say
that.
hi to Ray for me.
I will. And my TWiM guest is a microbiologist from NYU, and her name is Haran Darwin.
ah,
And she does really cool molecular biology of Mycobacterium tuberculosis. But of course, my first question is going to be, are you related to Chuck? I'm sure she gets that all the time. I don't know how common a name Darwin is. I guess it is. Wouldn't it be cool if she says, yeah, I'm actually a descendant?
that would be very cool. I feel like we would have heard of that, though, if
Yes.
one of his descendants was a scientist.
Indeed. All right. So today, what you want to do a monkeypox update? Rich?
I just slipped this in there because I was curious. There's, in terms of, following up on the discussions we've had previously about how it might spread, what populations it's hitting, all that kind of stuff. There's really no change as far as I know. But I found this CDC has a nice summary, and I linked to this global map that is tracking the cases. And it says that there are currently 1,019 confirmed cases globally. And I don't know. That does not seem to include any African countries, as Breanne suggested last time. And these are confirmed cases. So 29 countries, 1,000 cases, 300 in the UK, 198 in Spain, 153 in Portugal, 80 in Canada, 65 in Germany, France 51, Netherlands 40, US 30, Italy 20, Belgium 17, and then it goes down to less than 10.
Just one case in Morocco.
One case in Morocco, one case in Norway. So I just thought,
Yes.
that much of a status report is worthwhile.
So it's up from last Friday, which
Up from
was about
last Friday.
700. So that's a lot.
Yeah, and I would assume that some of that has to do with increased surveillance. Some of it has to do with incubation period following the initial outbreak, and if I'm kind of figuring that this is going to peak and die out. I hope, due, among other things, to people getting wise to it,
Yeah.
I hope.
So I would hope that would happen within the next couple of weeks, but we'll see. This gives us a baseline.
Yeah, they have a map of the U.S. with all the cases. California has the most. None in Texas.
How about that?
Yeah, none in New Jersey. We have 7 in New York State. Interesting. All right, we'll keep an eye on it. We have a snippet and a paper for you today. Snippet, I thought we'd talk about pets and SARS-CoV-2. This is a paper in Emerging Infectious Diseases, a publication of the CDC. It's called Risk Factors for SARS-CoV-2 Infection and Illness in Cats and Dogs. And I'm going to give you the bottom line. If you sleep with your pets and you kiss them, you're more likely to give them SARS-CoV-2, right?
Right. That, and I suppose I shouldn't be too surprised about this, but the seroprevalence in pets is in pets that are from households where there has been SARS-CoV-2 is quite high.
Yeah.
And I suppose I shouldn't be surprised at that, but I was.
Yeah.
Well, by quite high, you mean, say, 50%.
Yeah. To me, that's quite high.
Yeah. So the authors are, the first author is Dorothy Beansel, and the last author is J. Scott Weiss. They are from a variety of places. Ontario Veterinary College, Toronto Animal Services, University of British Columbia, the BC CDC. And so I like the first sentence where they say SARS-CoV-2 originated in horseshoe bats. Don't you love it? There's like no fooling around. Yeah, it's from bats. I like it. And they say, as we have talked about, other species have been shown to be infected, including household cats, dogs, ferrets, hamsters, of course, deer, mink, Syrian hamsters. So interesting. We didn't do this paper. I had my eye on it, but it was a report, I think, was it out of Hong Kong, where some infected pet Syrian hamsters might have transmitted SARS-CoV-2 to humans. White-tailed deer, of course, have been multiple studies showing infection of them and possibly transmission among deer.
Definitely transmission from mink back to humans.
Yes, from mink to humans. And the mink gets sick. They're highly susceptible to pneumonia, mortality rates of 35 to 55%. But in many other animals, they don't seem to be serious disease. They're subclinical or transient respiratory or gastrointestinal disease.
And that includes some of the zoo animals.
Yes.
Among others.
They don't know if the deer experience illness. I guess they haven't studied it enough.
I think it would be hard to monitor the deer for a long enough time to really see if they're ill.
Some of the deer are on farms, I think. There are actual. Didn't we have this discussion that there are actually deer farms? And I would think that might be easier to keep an eye on those guys.
I seem to remember there have been some infection of deer in the laboratory, but whether they get sick in nature is not clear. Anyway, this is about companion animals, which, of course, have very close contact with humans. And they say most data suggest that they're infected transiently, but the infection rate's anywhere from 0.2 to 43%. and maybe, again, subclinical or transient respiratory or gastrointestinal disease. So they wanted to look more closely at companion animals, pets, cats and dogs, and see if there are any risk factors for infection. So this was a study where they would have people who had a diagnosis of SARS-CoV-2 in the previous 3 weeks, they're pet owners, and they said, would you like to have your pet swabbed and tested? So enough people did to make this study possible. So you had to have COVID in the previous 3 weeks, and this was done from April 2020 to August of 2021. And the animals that were eligible were dogs, cats, and ferrets, any age, any clinical status. The only thing that ruled them out is if they were nasty. They say the only exclusion criterion was medical or behavioral issues that precluded safe sampling.
And they've got this a couple of sentences here really I found amusing. They talked about variations in the samples, and they said they cited as a source of variation, quote, the challenge of obtaining representative samples from the nose in cats.
Yeah.
I
can imagine.
I was imagining a few things in the challenge one might have with sampling some of these animals.
Yeah.
You need a pretty small swab to do a cat's nares, right?
And the cat is not going to go for this, I can tell you.
Yeah.
And cats, when they aren't happy about something, it can be pretty hairy.
Yep.
Yep.
All right, so they extract nucleic acid. They do quantitative RT-PCR. They're amplifying the N gene, and that they also do some serology, as you see. So we have 283 swab specimens from 65 cats, 49 dogs, and 6 ferrets, 70 nasal swabs, 90 oral, and 107 rectal. They have 16 fur samples. And of those, 5 cats and 1 dog had positive PCR results. So 5 is 7.7%, and 1 is 2%. So pretty low.
And presumably that represents an ongoing infection, or either that or a very recent infection.
That's right.
And they also specifically talk about whether these are the nasal swabs or the oral swabs, the rectal swabs, or the fur. And they don't mention the fur samples at any point when they're talking about positivity, which indicates to me that they didn't get any positive samples from the fur. I've actually had people ask me that question. If there was a household member who was sick, could petting their pet, be a fomite. And so at least based on the 16 samples in this study, they don't talk about any having PCR positivity.
A number of these they called non-negative because the CT values were there, but they were high, like 36 to 39, which is probably not infectious at all. And one cat was retested weekly 5 times and had positive results during the first 3 weeks.
And I'm glad they did that. So that's sort of internal confirmation that this cat had an ongoing issue.
And then they did whole genome sequences from 2 positive cats, and they could see that, yes, this is a lineage at the time that was infecting people. So this in the same geographic region as these pets lived. So it went from people to pets. Then they did some serology. They have serum from 59 dogs and 48 cats. This is now both households and 1 animal shelter. So it's a mix of sources. Seropositivity for IgG and IgM. This is first just ELISA, 42 to 62%. All IgM positive dogs were also IgG positive, but a quarter of the cats were IgG positive but IgM negative. I don't know why they're different. I guess the cats were farther from the initial infection, right, Brianne?
Potentially. I do not know enough about isotype switching in cats to know if there's something unique about cat IgM.
Yeah, because the dogs were both. You would not think the dogs were any different in how far they were out, but maybe there's a difference. Anyway, they did some statistical analyses to see if there are any correlations between being seropositive and some kind of behavior. They found an association between seropositivity and owner-reported onset of a new respiratory disease in dogs at the time of the owner's infection. So, the owner would have an infection, and then, hey, the dog seems to be having a respiratory problem, and those often turned out to be seropositive, but not in cats. There wasn't an association there. No significant association between seropositivity and owner-reported onset of new respiratory disease in the pet at the time of owner's infection, but they did observe a significant association of seropositivity and owner-reported new onset of clinical respiratory, gastrointestinal, or systemic signs such as lethargy in the pet, and that's for dogs and cats. No association between time the animal typically spent per day with the owner. However, cats that spent less than 2 hours per day were less likely to be seropositive. I guess if you're at work all day, right, you're less likely to infect your cat. They didn't see the same results for dogs. No correlation with the number of people in a household.
There was a correlation with cats sleeping on the bed with the owners.
Yes, for cats, association between positivity and being kissed was significant, and for sleeping in the same bed, right? Yeah. For dogs, licking hands or face of owners was associated with seropositivity. Also, for dogs, there's an association spending 19 to 24 hours with the owners.
So they've got two tables here. One that reports the statistical analysis for results that were judged positive if the OD in the ELISA, the readout in the ELISA, was greater than 3 standard deviations above the mean of negative controls.
Mm-hmm.
And the other is basically the same data, but counting as positives results that were greater than 6 standard deviations among the mean of negative controls. And, the significance for various covariables comes out to be a little different depending on what you decide is a positive result.
Yeah, that's right.
And this triggered in me a memory of a time in my lab when I was co-mentoring a student whose other mentor was in the vet school. This guy was a veterinarian working on his PhD, and the problem, the vet that was his primary mentor was well networked, and people would send him problems, okay? So this problem came from the Marine Mammal Center in the Marin Headlands, north of San Francisco, where they had rescued seals. And the seals were getting poxy-looking lesions. And they were concerned that the seals were catching it in their facility. So they wanted to know what this was. And they put this student on, and this student, Hendrik Nollens, was brilliant. And he actually, nobody had characterized this virus before. And I reviewed the literature today, and I'll get around to why here in a second. I reviewed the papers that came out of this today. And he actually, did the histopathology on the lesions. He identified a pox virus. He actually grew it in culture, isolated it, purified it, got nice EMs of it, created an ELISA using whole virions. And his positive controls were diseased animals. His negative controls were aborted fetuses.
Of seals?
And then he did excuse me?
Of seals?
Yes.
Human? Okay.
Yes, aborted seal fetuses. And, this is clinical material, right? From probably, I don't know. Well, I don't know. I don't know exactly the source of that. But, and then the question was, what's the distribution of this in nature? The answer was that these viruses, it turns out to be a parapox virus, and in nature, about 90% of the animals are seropositive. And the infection in the facility was probably a recrudescence of a persistent infection, which put the minds of the facility owners at ease. It involved also a bunch of swabbing of the facility, et cetera. Great studies. But cool. The deal was, okay, when you do the serosurvey, you get a range of values. How do you decide what's positive?
Sure,
you've got a negative control, you've got a positive control, but where's the cutoff?
Yeah.
And they spent a lot of time with this. And this reminded me of it. I think that's probably why they've got these two different tables of 3 and 6 standard deviations above an average negative. Okay, because you may get they're saying, well, you could draw the cutoff here or you could draw the cutoff here, and this is what the impact is on what you consider on the covariables. In the study that Hendrik did, I had to look this up again. They used, I'm wondering if either of you are familiar with this, they used a statistical method called receiver operating characteristic. Are you familiar with that, Brianne?
I learned that in biostatistics, but I could not tell you. I remember ROC curves, but I am not sure I can tell you much beyond that they're called ROC curves.
Right. It was way over my head. But at any rate, it was a way of computing a reasonable cutoff for the ELISAs. But my point is that this is not straightforward. Okay. And my guess is that this is why they have these two tables using these different values for positive controls.
They also did surveys of cats from neuter clinics, and they've again found up to, looks like on this table, like I said, around 16%. They found IgG in about 16% of these clinic cats. And then they had some cat and dog samples from Humane Society. The 67 cat and 8 dog samples, 9% overall were seropositive. They say basically compared with cats originating from households, those that were in a shelter, rescue, or foster facility were 3.6 times as likely to be seropositive, which makes sense, I suppose, right?
I was confused by that, I guess.
I think because there are a lot of different people are working in there, as opposed to one owner, right?
Right. So. I guess for some reason I was imagining those cats as having spent little time, the cats in a shelter being just coming into a shelter, and so really having spent very little time around people. But you're right. If we imagine them in the shelter for a while, they've spent time around a lot of people. So
that makes sense. I was with you, Brianne. I was a little puzzled by that.
So animal source at this neuter clinic. So this is positivity. Household pet, 8%. Shelter, rescue, foster, 23% positivity.
Okay.
And then feral cats, 19%. So that's high also. That I don't understand because they're running around, right?
Exactly. That's the one where I was a little confused, is that the feral cats, I would expect would have a harder time getting infected.
Maybe they're feral cats that are in the shelter, right?
Right.
I don't know.
Maybe
these guys need to do a receiver operating characteristic analysis.
Maybe they do.
Maybe the feral cats were very stressed. And thus, had reduced immune responses because they didn't have someone cuddling them and giving them good food and treats.
So the bottom line here is that transmission of SARS-CoV-2 from humans to pets is common, but mostly they don't seem to get very sick. So people who have been worrying about that, we've gotten a bunch of questions. Someone asked me the other day, are they going to make vaccines for cats and dogs? I don't think so, because it's not a serious illness.
And there's no information in this or elsewhere, as far as I know, about whether or not there's spillback from these animals to humans.
Yeah, no information, no.
That would be really hard, because how would
Yeah.
you say that, your household cat was where the source of the spillback instead of your other household member?
Unless there was some sort of genetic tracing you could do.
Yeah,
you know.
that's true.
Yeah, with the minks, the change occurred when it infected minks, and then you could see that when it went back into people, that same amino acid change or whatever. But so they cite a previous study of experimental infection of kittens with 1 million TCID50s of virus intranasally and orally. They had viral RNA for 10 days in swabs. They had subtle pulmonary lesions, but they cleared the infection. It wasn't lethal, and they were okay. So, and that's a lot of virus, 1 million.
As I recall, when we quite a while ago, when we looked at the infected lions, or where were they? A zoo somewhere. Was it the Bronx Zoo? I forget, but there were a couple of infected lions, and one of them at least was
that's
had
right,
a cold.
Bronx Zoo. Yes, that was the Bronx Zoo, and then the gorilla was in the San Diego Zoo, right?
Right.
So they make a note in the discussion. The prevalence of seropositivity was moderately high in cats with no known exposure to infected people. I guess those are the feral cats, right?
Right.
Mm-hmm.
Yeah, so the lack of metadata makes this challenging to interpret, because it's possible that cats from the Humane Society or a neuter clinic had previously been exposed to infected humans.
Yeah, I also wondered, there is at least one other feline coronavirus known, right? Although it's not super closely related.
Yeah.
And so part of me wondered, well, are cats infected with other coronaviruses that may impact their immune responses?
Is there any cross-reactivity?
I don't know.
They don't mention it at all here.
They don't mention it, and I think that other virus is maybe not a beta coronavirus.
Okay.
If I remember correctly, it was a little bit further away on the phylogenetic tree. And I also don't remember about the pathology. So I think that one might cause some disease in cats. So perhaps we would know if the cats had previously had that. I don't know. But I'm guessing we maybe don't know about all the coronaviruses that exist as well. So there could be another one.
Feline coronavirus is a common viral infection in cats, generally causes asymptomatic infection, but can cause mild diarrhea. As yet poorly understood, changes in the virus can give rise to mutants that lead to the development of feline infectious peritonitis. We've discussed this in the past.
That's the one that I was thinking of.
Which is a nasty disease, as I recall.
Mm-hmm.
But it certainly would have been
alpha.
a good part of the discussion to throw that in there and ask whether there's any potential for cross-reactivity in the serological assays.
All right, conclusion, cats and dogs can be affected from you. They don't seem to get very sick. Risk is low for them. Unknown cat-human transmission or dog-human. Don't worry about them. The risk factor is kissing and sleeping with them. So there are other papers that look at similar things, but I think that's a good example. All right. Today we have a paper about a DNA virus. This is in eLife, a novel lineage tracing mouse model for studying early murine papillomavirus infections. MMUPV1 is the abbreviation of the virus, mouse-specific papillomavirus. And this is from the University of Cyprus. How about that?
Cool.
Let's see here what we have in terms of... all right, the first author is Vural Yilmaz, and the last author is Katerina Strati, which is the grad student that was with Paul Lambert.
Senior author, Katerina.
Katerina, cool. On Cyprus. Very cool. Rich, you want to give us a rap about polyomaviruses, human papillomavirus? Yeah.
Okay, I'll try.
You used to work on polyomas, right?
I used to work on polyoma, but I have a long-standing fixation on papillomaviruses because I've given this rap before. I think my sister-in-law died at the age of 34 from a cervical carcinoma.
Right.
In 1992, I think, and I taught papillomaviruses to the medical students for over 20 years using her as my clinical case. And watched, so it made it for a compelling lecture because I had this rap about her and her courage in dealing with all of this.
She was a doctor, right?
She was a rheumatologist, at the Beth Israel. So, yes, small double-stranded circular genome DNA viruses, small being about 8 kilobases, I believe. That's enough for about 8 genes by my sort of thumbnail calculation. Naked protein shell capsid. Notable for, primarily they cause warts, right? Skin infections of one sort or another. As the further we delve into this, the more we realize that we are crawling with these things, and many of them don't ever cause any symptoms at all. But the ones that we're aware of, and there's well over 50 or 100 different types, typing based mostly on genome sequencing, but turns out to be relevant serologically. The different types cause different sorts of warts in humans, and of course there's papillomaviruses of essentially all over vertebrate species as well. They pretty much stay put in a species. They don't cross species boundaries very well, but within a given species, there's a lot of variation. And interestingly, the variation in terms of warts is there are different types that cause warts of different morphologies, specifically in different tissues. Okay. So there's a lot of specialization, apparently. The nasty ones, and there's a handful of really nasty ones, can cause cancers, where essentially what happens is, so these, if you've only got 8 genes, you're very dependent on the host to do things for you. And so what's typical in that sort of situation, I'm anthropomorphizing here, but what's typical in that sort of situation is the virus will encode functions that induce the host to make factors that are used for the actual replication of the viral DNA genome. And a consequence of that usually is to kick the host itself into synthesizing DNA. So you get unscheduled DNA synthesis, which translates into hyperproliferation of cells, which translates into a tumor. Okay. And in the case of warts are a tumor. It's a benign tumor that usually is taken care of immunologically, resolves, and goes away. Just a few episodes ago, I had that essay on warts that suggested that you could hypnotize them away. You have to look back at that one. But what can happen in particular with, so one of the sort warty things that can happen is a flat wart on the cervix in women. And quite often that, and that's caused by a papillomavirus. Usually that just resolves spontaneously, and that's okay. But there are several high-risk viruses where that progresses to a cancer. And that's the cause of cervical carcinoma. Think Henrietta Lacks, HeLa cells. And it's important to understand in that case that once you've progressed to a carcinoma, you're no longer doing virology, you're doing oncology, because what happens is that by accident, really, the viral genome gets linearized and integrated into the cellular genome. So the virus is essentially dead, but now you have a permanent copy of the virus genome in the cellular genome, and that can include these genes that cause unscheduled DNA synthesis. So now you have a cancer. You got genes in the cell that are causing the cell to proliferate abnormally, and that can progress into a malignant cancer.
I remember when they did the genome sequence of HeLa cells, they saw the E6, E7 integrated right into chromosome 18.
Yep, intact. And so the cool thing is that since we know now the viral etiology, and it's not just cervical carcinoma, there are some other cancers, some anal cancers, more and more head and neck cancers, and et cetera, both males and females that are caused by a subset of these papillomaviruses. Now that we know that there's a virus associated with these cancers, we can make vaccines. So there are papillomavirus vaccines available that are essentially anti-cancer vaccines. And this is not a trivial amount of cancers. In this paper, they talk about the papillomaviruses accounting for up to 5% of human cancers. So that's not at all trivial. I think the other thing to understand is something about the biology of these things. The epithelium, we're talking about what they call stratified epithelium. Can be either regular epidermis or it can be a mucosa. They have fundamentally the same structure where you have a basal layer of cells that are essentially stem cells for the rest of the cells in the epidermis, and they divide and give rise to cells that migrate up through, I don't know, somewhere between, I suppose, I'm just guessing here from what I've seen, a dozen and 20 or 25 layers of cells in the epidermis. And these cells migrate up through the epidermis and they differentiate as they migrate up to ultimately giving you this keratinocytes on the top that are these cells that are full of keratin that are in the very upper layers essentially dead and form this hard layer in the epithelium. Mucosa is somewhat different, but the stratification and the differentiation is very similar. And what these viruses do that's very interesting is that they infect cells in the basal layers of the epithelium through a wound of one sort or another by mechanical transmission. And the genome travels to the nucleus where it resides and replicates at a low level as an episome, that is a circular piece of DNA separate from the chromosome, and makes some very early gene products, and then but doesn't make any virus at that point. And then as the cell migrates, as that infected cell migrates up through the epithelium, the differentiation signals and changes that happen normally in that cell trigger changes in gene expression in the virus, so that now you get expression of these proliferation genes, other early genes, as you move up. And ultimately, in the top of the epithelium, the differentiation signals trigger the expression of the late genes, which includes the capsid proteins, so that now you make mature virus, which is then sloughed off in the very upper layers. Importantly, what that means is that the biology of the virus replication requires all of this differentiation. You can't just take a cultured cell and infect it because it doesn't have the right differentiation state to do all of this stuff. And in the past, what has been done for in vitro culture is to create these artificial epithelia in what are called raft cultures, which are very tricky to do. And then you can infect those and you can reproduce the whole biology, but you can grow virus, but not much. It's just enough to sort of see what's going on, to convince yourself that you've reproduced it. So all of this analysis has been done by recombinant DNA technology. Okay. You can't grow virus in culture. And so good animal models for this, or something resembling, or a culture model, are quite valuable. And this paper describes, for the first time, a mouse papillomavirus, which has potential for, actually, the virus was discovered in a previous publication by another group, but this does some work on it. So now that we have a mouse papillomavirus with some described characteristics as described in this paper. Perhaps this provides a model for looking at papillomavirus infections in a laboratory, homologous laboratory, paired host pathogen model.
Yeah, the guy, one of the main players who made the connection between these viruses and cancers was Harold Zurhausen, who got the Nobel Prize for that. And I interviewed him a number of years ago. I'll put a link to that in the show notes. So the problem is that there are some animals, but they're big animals and they're not mice, right? So that's why this is interesting, because, of course, mice are a convenient, small, easily reproducible, reproducing animal model. So I went and looked. Go ahead.
No, I was going to say, I think that the other strengths are, of course, that mice are more genetically tractable,
sure.
So that you can actually start to interrogate the mouse. And also that mice are organisms where there is also some skin biology that has been done, because this involves things like needing to make a wound to get to that basal layer, and needing to think about all of those layers of skin. There are certainly some other animals whose biology may be less characterized in that way.
So the virus was discovered in 2011. I found the paper, Novel Laboratory Mouse Papilloma. So the name that I called it, MMU, is mus musculus, then papillomavirus 1. But in this original paper, which came out of India, essentially, a mouse colony in India, he said we had mice. Lab mice are carefully cared for. So, infectious diseases they say they're rare, considered all but eliminated, but that's not true. Many mouse colonies have infections, and you can just monitor for them. But they noticed a spontaneous development of what they called florid papillomas. They have a picture of one mouse, and it's a florid papilloma.
Florid indeed, yeah.
Mm-hmm.
Yeah.
Wow. Reminds me, the last time we talked about papillomaviruses, I think the episode image was the
rabbit.
Shope fibromavirus.
Oh, yes, the rabbit, the rabbit with the papillomas.
Rabbit with the big, these warts can get huge. Okay,
jackalopes,
yeah.
Now, these are nude mice, which is probably why they got this infection, right?
What are the immunological characteristics of nude mice?
So nude mice have it's a FOX transcription factor deficiency. Although they were found naturally. And the key thing that's important immunologically is that they don't make a thymus. So they do not have a thymus, so they have no T cells or T cell dependent responses. It also so happens that this transcription factor changes mesoderm development, so they don't have hair as well. But as I have to remind my students often, the hair and the thymus are actually not related.
So if they don't have T cells, that's going to impact on their B cell response too as well, because they don't have the helper cells.
Exactly.
Right?
So they'll have some B cells, but the B cells won't make their full range of B cell responses because they're not getting help.
You say these happen naturally. You mean spontaneously in a mouse colony?
Spontaneously in a mouse colony.
Okay. Well, so I'll tell you, this mouse doesn't look happy at all.
They found the mice with these florid papillomatosis. They sacrificed all the mice so it wouldn't spread, but then they did some immunohistochemistry. They did some, they extracted DNA and they amplified it, and they said, this is papillomavirus. And then they injected cell-free extracts into other mice and got papillomas. And so that's the virus that they're working with in this paper. And so the model is very interesting. So what they do is they have a mouse line, which where the gene encoding yellow fluorescent protein has been integrated into the mouse genome, but it has a stop codon before the gene that needs to be removed by Cre recombinase. So the Cre-Lox system is a system from bacteria where the lox is the site that's cut by cream cheese and lox. You can remember it that way. So the lox is the sequence, and the cre is a recombinase, an enzyme that will cut it. And it's very useful if you want to cut something out. So in these mice, they're not going to be yellow. The cells are not going to be yellow until you put cre recombinase in, and then it will cut out the stop codon, and then the mRNA can be translated to protein and make yellow fluorescent protein.
So the lox site is basically 2 bits that flank something, and the recombinase interacts with both those sites and clips out what's between them. And the repair leaves, does it leave a little bit behind?
Trying to remember it right now. I should know this.
If it does, it does it in a fashion where you can, where it doesn't necessarily interrupt the sequence that you can do it without harming the sequence that you're interested in studying.
You do it without harming the sequence.
And then so that's the mice, and they breed these mice, so they have stably integrated this stop lox yellow fluorescent protein gene. And then they have the viral genome, the murine papillomavirus genome on a plasmid, where the viral genome is interrupted by cre recombinase flanked by lox sites. So when you put that in cells, the idea would be that the cre recombinase is made, it cuts out itself. The plasmid can now be ligated in the cell and reconstitute basically the viral genome. The cre, and then whatever cell that goes in, the cre recombinase is now going to activate the yellow fluorescent protein gene.
Brilliant.
Isn't that cool?
So it's the originally infected cell has the cre, but not sort of later cells infected by progeny virus.
And as the cells differentiate, right, so that you infect a basal cell, presumably, as they differentiate, they're going to be yellow. So you can track the virus infection progressing. You could look at the yellow, and you can also look for viral transcript as well. So they designed this plasmid. In the first few experiments, they test it. They have cells in culture, mouse embryonic fibroblasts, MEFs, and these MEFs were from the mice with the yellow fluorescent protein gene that's silent because it has lox flanking a stop codon. And they put in the viral plasmid, and the cells turn yellow by immunofluorescence, as you would predict. And then they can extract DNA and do PCR and show that, in fact, the cre lox is gone and the viral DNA is now intact. So you could, in theory, you could initiate a virus infection cycle with that plasmid, right? Okay, so you put the plasmid in cells. The plasmid that you put in is transcribed. The RNA makes cre. The cre protein cuts out the lox from the DNA, cuts out the cre gene. The cell ligates it back together. The cell doesn't like to have DNA ends, right, Rich?
Right.
It likes to ligate them back together. And then you have an intact viral genome. So then they look for viral transcripts. There, as Rich said, there aren't that many. They can find viral transcripts, early and late transcripts in their transfected cells. They have a control cell experiment where they don't see it. So it works. Constructed plasmid can turn on the YFP in the cells. It excises the cre, and the genome circularizes, which they can tell because they're making the right viral transcripts. E1, E4, L1, and E6 E6 would not be made if the cre was still stuck in there. That's cool, right?
Yeah, very cool.
It's very cool.
I like that very much. So that's in cell culture. So now they go, next we use the plasmid in mice. Now they say infection with this virus, MMUPV1, is mainly cleared by CD8 T cells. So they say, well, we should immunosuppress these mice. So they transiently immunosuppress them by UVB irradiation, which suppresses adaptive responses and leaves innate immunity unaffected. How does that work, Brianne? What does it do?
A lot of it has to do with hitting the rapidly dividing immune cells. So the B cells and T cells that are rapidly dividing are going to get mutations from the UV light and then divide quickly and not be able to repair their DNA. So they're going to undergo apoptosis.
So they don't actually show you with and without irradiation. I would have been interested to see the effect, but they say, okay, we're going to irradiate these mice, and then the base of the tail skin is abraded with a pipette tip. So it's a plastic pipette tip, right? So it's not a lot of abrasion, but it doesn't take much to get to the basal layer, right? Which Rich was saying, those are the cells that are infected normally, but we're not infecting, we're putting plasmid in. They deliver the plasmid to the skin, and then they harvested it at different time points to look to see for yellow fluorescence by immunofluorescence. And in fact, they see clones of yellow cells in the skin, which they say is indicative of heritable yellow fluorescent protein activation in a skin basal cell. So I guess, Rich, even if the DNA were taken up into various cells, only going to start the cycle in the basal cell, right?
Yeah. At least that's the theory.
Yeah.
And the nice thing about this is that's what it shows. Now, I'm one thing that I'm puzzling over is the control. First of all, they have nice pictures here where they use 3 different fluorescence markers. One is a Herx stain, which just stains for DNA. These are sections across the from the tail after the infection at day 0, 3, 5, 7, and 10. And they have Herx, which just basically shows you where the nuclei are. Then they've got K14, which is a keratin. And so that basically marks epidermal cells, I think. And it looks like it is throughout the epidermis, that particular keratin. Some of these keratins are quite specific for different layers of the epidermis, but this one looks like it's spread throughout. And then they have the GFP marker and the merge of those. Now, they have this nice little diagram that shows what the hypothesis is, which is that you would infect a basal cell, and that as it migrated up through the epithelium and divided, that you'd get laterally more and more cells because of the division. So you get a sort of looking at a cross-section of the epidermis with the basal cells on the bottom, you get sort of a pyramid on its head, or a sort of a conal distribution, and that's what they see, though it sort of diffuses out at later times. Now, what interests me is they've got a control here that's CAG Cre, a different plasmid that has the same Cre in it, and they do the same experiment with it. And basically, I see the same thing, right? So it looks to me, then they don't comment much on this, but it looks to me as if that thing must, because you see the same sort of, unless you disagree with what I'm seeing, I see things that look like kind of conical lesions as well that progress in the same fashion. So, I mean, that doesn't invalidate what they're doing, but it suggests to me that maybe that plasmid itself is either preferentially taken up or preferentially maintained in cells that originate from the basal cells in the epithelium.
Yeah,
which would make sense to me.
I kind of get this, that they're saying that this method allows them to sort of lineage trace from an original basal cell up through the other layers. And so it doesn't matter if it's an infection or not. If you get Cre into that basal cell, you're going to lineage trace.
Right.
And so they're showing us that, okay, so believe our lineage tracing, it's not virus dependent, because now we're going to start to tell you in future parts of the paper about what the virus is doing.
Right.
But no matter what, if it was the virus or not, we could lineage trace.
Right. Okay. So that makes sense to me.
I'm looking at this and I have a former student who is also a TWiV listener who I know for a fact will contact me after this episode. I can already imagine our conversation. But she has actually worked quite a bit on mouse skin. And so I'm already thinking about all the questions that I'm going to ask her about. And I really want to know right now why they're using tail skin and how much of this might change with skin in different parts of the mice and why they chose the tail.
Good point.
My first thought is because it doesn't have a lot of hair on it.
That was my first thought too.
Have to worry about getting rid of the hair.
Yeah.
But I wonder whether different parts of the mouse's body would have different things going on with the skin.
Yep. Okay. Anyway, they put these.
By the way, these mice. I'm forgetting here. These mice are immunocompetent, or are they not?
So they were treated with UV.
Oh, that's right. UVB. Okay.
To immunocompromise them, but otherwise they are immunocompetent.
They're genetically competent. All right. So they introduce the plasmid into the skin. They harvest, and as we've seen, we see characteristic clones indicative of activation in a basal cell. So they say here we have a system where we can see the cells that have the virus and their progeny over time. And the control that Rich said, the CAG Cree plasmid, doesn't encode any viral genes. So you get a similar pattern, but there are no viral gene expression, as you'll see. So then they look for viral genes and transcripts. They use RNA in situ hybridization. And this is RNA scope, Rich. I don't know. You remember we talked about this from your colleague at Florida, right?
Dave Bloom.
I'm thinking of Stephanie Karst, who used to
Stephanie Karst as well. Yeah, I was first introduced to this by Dave, but of course, they're talking to each other all the time. It was Stephanie Karst that used this to look at mouse norovirus infections.
Remember, she would roll up the intestine and slice it.
Yeah,
and you get the whole thing, and then you do an RNA scope with many different colors. Anyway,
so this is sort of in situ hybridization on steroids, right?
Yeah.
It allows you to look at, it's very sensitive. So you can see almost single transcripts of a given RNA in a cell. So it's very sensitive.
So they find at day 3 after putting the plasmid in the tail, they see E6, early region 6-7 transcripts. And then early region 1-4, 5 days onwards, peaking at 10 days. Of course, none of those are in cells that had just got the CAG Cree plasmid, since there's no genome in there. And they could tell that it's viral RNA and not DNA by treating sections with DNase or RNase. So this is RNA signal. So the plasmid is getting in, the Cree excises itself, and then the genome can be transcribed to give these viral mRNAs. Then they swab the surface of the tail. They want to find the genome on the surface, right?
Yeah, because they've put it at the bottom, so they want to show it goes all the way up.
Yeah. Which is, to me, I'm not quite sure how this is working, because if you scrape, you've got that open wound, right? Although maybe by so many days it's healed already, right? I don't know.
Yeah, I'm still unsure exactly how much of a scrape you're getting with a pipette tip to know how long that would take to heal.
Yeah. Anyway, they swab the surface of the tail and they isolate DNA from that swab and amplify, and they can confirm that the Cree cassette is gone, right? So what's making it to the surface has lost the Cree, which is what happened after you put the DNA into the cells originally. And the L1 gene is there. They can do that by PCR. And L1 is a late gene that's away from the Cree site. So it is actually the version of the plasmid they put in without the Cree gene.
And the L1 is one of the capsid proteins. So that would be basically diagnostic of a cell that's making capsid proteins and potentially making virus.
Yeah. So they say with the presence of L1 confirms the viral genome is on the skin surface. So it's L1 plus not having Cre, right? Because now you have the viral genome.
So that would be consistent with this whole thing I was describing before as the infected cell traveling up through the epithelium and getting maturation of the virus in the upper layers of the epithelium.
And they are saying that they're detecting this on the skin surface at days 5 and 10 following deposition. So that might be long enough for it to heal.
It could be.
You've also got to assume it's got to be enough time for that differentiation to occur.
Yeah. So they say the presence of the recircularized viral plasmid on the surface could be a result of the migration of the progeny cells to the upper layer from the basal layer, as they say. And then they're assuming that the viral particles are shed on the very top cells, as Rich said before. And so they actually looked by transmission electron microscopy in sections, and they could see virions. And if you look at the picture, in the perinuclear area, so there's a lot of virions there. I don't know if they're the right virions. I assume they are. So the control plasmid, you don't see the virions. The CAG-Cre plasmid, there's no virions, and their plasmid there is. So it's not just virions that are present in these mice, right?
So this is what got my attention and prompted me to write Paul Lambert, who's a papillomavirus expert at the University of Wisconsin, because I wanted to know more about the virion assembly, and I looked back at the original isolation paper, and it has the virions in the nucleus, as if capsid assembly goes on in the nucleus.
Yeah.
And so I wrote to Paul, and not only that, but it's interesting to point out, I think it's interesting to point out, that this virus causes a cytopathic effect, or papillomaviruses. I don't know if it's generally, but this virus, apparently, according to the discovery paper, the original discovery paper, it can do this in those nude mice, causes a cytopathology where the affected cells are called koilocytes, where the nuclei kind of shrink up and there's a lot of space around the nucleus, and they're very characteristic looking. It's the virus causing this. And in fact, it's the presence of koilocytes that the pathologist is looking for in a pap smear initially. And they can, if they see that, then they can further go on now and look for virus genome and even type it and all the rest of that kind of stuff. But so I asked Paul, wait a minute, these guys are seeing virus in the cytoplasm and though close to the nucleus, and the report of the isolation has it in the nucleus. So where does this happen? And he wrote back and gave me a couple of papers that also have virus capsids in the nucleus. He complains that in both his own work and the work of others, that there needs to be confirmation by immunogold staining to show that those are really virus capsid. But he says, sure looks convincing to me. So I wonder what's going on here. And I even wonder whether, first of all, they don't report koilocytes. Matter of fact, I think they specifically say that they didn't see koilocytes in this mouse model. And I wonder whether there's maybe something funny going on with this section, some sort of a fold or topological thing, and this isn't actually in the cytoplasm, or I don't know how these things get out of the nucleus and out of this, except maybe they never do because the cells slough off, right? So I got questions, put it that way.
Yeah. Well, so I think there's one other piece that is in this part of the paper that's sort of a little bit related to what Rich is talking about in terms of the cytopathic effect, because there was this one control that they mentioned that I thought was really clever. And I was really impressed by that comes up here because they also, when they're looking at the skin swabs, they do some swabs of the skin at day 30 post infection. And at day 30, they don't find viral genome. And you could imagine there being two potential reasons for that. One, perhaps maybe the cells have stopped making virus. The infection has been cleared in some way. Or two, maybe the cells that were originally making the virus died. due to cytopathic effects. And I hadn't thought about this when I was looking at the model, but I just really liked this experiment, that they could actually look to see whether those original cells that they put plasmid in were still present or not by looking to see if there were still any YFP cells.
Right.
So were those original cells present, or their progeny present, or have they been replaced by cells coming from other parts of the basal layer? And so they could still see YFP positive cells. So it's not all cytopathic effects. Some of it is actually clearance. And again, I hadn't thought about the fact that you could do that, but I liked it that they did.
Interesting.
I mean, when these viruses are triggering cancers, it's an abnormal event. It's a rare event, right? So I guess in most cases, the cells are going to either slough off or the infection is going to be cleared by CD8s in some way. So I'm not sure how often you might expect to see transformation in this mouse model. That's something they could look at in the future, right? Would be interesting to look at that. Is it 1 in 100 mice or is it more? Whatever. Anyway, they do a couple more experiments to take advantage of yellow fluorescence, and that is they do some flow cytometry. They put their plasmid into the tail, then they dissociate the cells and do flow cytometry, and they can look at kinetics of yellow fluorescent protein. They see, at day 5 the number goes up, and then it starts to decrease at day 10, and then day 30, as Brienne said, there's still some there, but it's decreased. And then they look for a proliferation marker. This is a protein Ki67 marker of cell proliferation, which is, you'd expect the viral gene products to induce proliferation, as Rich said, so that the DNA synthesis apparatus is available for the viral DNA. And this marker Ki68 goes up around day 5 in these YFP positive cells, which is cool. So the cells with the viral plasmid in it have higher proliferation, as you would expect. Then they look at MHC1 expression. So it's been reported by others that infection with papillomaviruses decreases MHC1, but as they say, this has only been done in cells in culture. So let's look in mice. So they do their delivery of the plasmid to the mouse tail, and then they use flow cytometry to look at both MHC1 and yellow fluorescent proteins. At day 5, there's lower MHC1, and in the control, actually, they see increased expression, which they say may be a consequence of YFP protein, right? I don't know if that's a reasonable idea or not, Brienne. What do you think about that?
Potentially. I think that I was just looking at some of the methods surrounding this part of the paper, because I think that technically this is a very challenging thing to do. Getting the cells out of the skin and running them on a cytometer is not an easy thing to do, and those cells are going to have a fair amount of background.
Yeah.
So I'm still sort of at the, oh my goodness, look at the cells running, skin cells running through the cytometer point. And so, yeah, sure. I could imagine a couple ways that you could have the YFP leading to that, but part of it is just great job getting skin and running it this way.
We don't actually see
I'm happy to hear you say that, because I was wondering about I've never done flow cytometry, so I don't know, but I could imagine there being issues.
Yeah. You know, the traditional blood, spleen, things like that, those are pretty easy. But when you start getting into tissues, it can get very tough very fast. I wonder if
did the UVB treat the controls? I think they did, didn't they? I wonder if that would do anything.
They should have, I think.
I think they did.
Yeah. But they say, there's a new antigen here. Maybe that's part of it. I don't know. But they don't actually show the plots, Brienne. They just show graphs of the data. I don't know. What's the common practice? Should you always show your plots or what?
It really depends. I like to see plots.
Yeah.
But I can imagine with plots like these, they would have to have a lot of negative controls that they used for their gating.
Yeah.
And my guess is that when they showed me the plots, my first response would be, oh, I want to see those controls. And, oh, I want to see this. And at some point, they can't show me their entire flow cytometry library. So I guess I kind of have to buy what they're telling me here.
So they conclude that maybe the virus decreases expression of MHC1, and that's an immune evasion strategy. So that's something they could work on more in the future. Although I don't know. I mean, what now? MHC1 is on all cells,
all nucleated cells.
Okay,
and it's presenting to the CD8 T cells. So it does have that nice sort of relationship with CD8s being important that they talked about earlier.
Yeah. So I guess that's something they could follow up in more detail. Yeah. Although they're not, the problem is they're not an immunology lab, right? The PI is a virologist trained as a papillomavirus person. So they're going to have to get some collaborators, I think. Otherwise, they're going to run into problems at some point. So those are the data. The virus delivered in this way with a plasmid causes proliferation of cells. Maybe MHC1 goes down, and they say we have a system for lineage tracing, which maybe we can use. They say can use to answer a variety of outstanding questions. And I would have liked them to list some of them because the discussion is like one paragraph. And like, what are the outstanding problems? Because I'm not a papillomavirus person. I don't know what they are. What are they? I'm surprised that their discussion is so short, right?
Well, they have their results section is called results and discussion.
Discussion, I suppose.
Then they have a conclusion section.
Yeah, I mean, I think about this and I start to think about some of the open questions in skin immunology. And ways that it could be used to address some of those.
But on the other hand, I thought, well, maybe they don't want to give away all the work that they want to do, right? Anyway, I thought it was a cool paper, nice technology, and worth talking about. All right, let's do a couple of emails. Brianne, can you take that first one?
I can. So Glenn writes, TWiV team, I recently got my second booster Pfizer, having had the original J&J and then a first Pfizer booster in November. Got the shot Saturday afternoon and rested the rest of the day, but felt okay and felt fine the next morning. I am not a health nut, but I do work out every morning, and Sunday is my bike ride workout. Feeling no ill effects from the vaccine, I took off for an hour ride of about 12 miles. In the shower, I am soaping up my left armpit, shot in the left arm, and it is like, whoa, egg-sized swelling. After the shower, I show my wife and assure her that this is a good reaction. I am proud of my response. By midday, the swelling goes down, then disappears, and I think, okay, the vaccine has done its job, and now my germinal centers are resting. Next morning, no swelling, and I feel fine and go out for a 4-mile run. Back in the shower and repeat. Big swelling again. I say to my wife, I think my workout is stimulating antibody response. She feigns interest and goes back to what she was doing. Midday, swelling is gone. Tuesday morning, body toning workout, shower, you guessed it, swelling is back. So 3 consecutive morning workouts, 3 swelling events. Wednesday, I have my usual elliptical workout, followed by a quick 1-mile run and no swelling this time. So I am curious as to if anyone has ever studied the effects of a good workout in conjunction with an effective vaccine to stimulate antibody production. Sure seemed to be some connection there. Could be an emerging field of research.
Brianne, before you go on to the rest, if it indeed was a lymph node, it shouldn't go down within the same day, should it?
Maybe if the workout is pushing a lot of lymph through the lymph node, you could have something really transient, but I don't think it would go down quite that quickly if it was a germinal center.
Okay.
P.S. Love the show. I am same age as Vincent, born 1953. For Kathy, our daughter just graduated from MU with her master's in information technology. Go Blue. We love Ann Arbor and going to be up there in July. For Rich, my wife can trace her ancestry back to the Gotchers, Supposedly, Samuel sold his 4,200 acres, and now the town of Austin sits on most of it. Alan, Dixon, Brienne, Amy. I got nothing. Quick joke. Which Paul McCartney song invites in a toxin? Brother Michael, Auntie Gin, open the door and let him in. Yeah. Haha, Glenn.
Great.
So what do you think, Brienne?
Yeah, what do you think, Brienne?
What does exercise do anything? You had a good idea, the movement of the lymph, I guess.
Yeah, I think it might change lymph movement in general, and that could be a good thing for immune responses. I don't know if it would be antibodies specific. And I don't know enough about the literature on it.
So it could be that it's not specific to his nodes where he got the shot, but that's the only one he was looking at. I mean, the armpit nodes are easy to find, and he maybe didn't look anywhere else, right?
Right. I mean, I guess maybe it was also interesting if it was only on the left and not on the right. That might give him some credence. I will admit I have had someone try to argue about the increased lymph being good for your immune response by strenuously trying to tell me that laughter was good for your immune system because it moves the diaphragm and that moves the lymph. And they were pretty sure about the immunology of that. I was a little less sure, but I'll do all the laughing I want to see if it works.
It's good. Rich, can you take the next one?
John writes, my name is John Sorrow. I'm a junior studying biology at Caltech and taking BI 115, viruses and applications to biological systems. I was listening to TWiV 903 and had two questions. Is this one of these class assignments?
Yeah.
Vincent? Yeah. Okay, fine. One, were there any hypotheses as to why total casualties from Omicron in just an 8-week period were so much worse than 23 weeks of Delta? I'll come back to that. Two, what are the possible mechanisms of COVID infection relapse? How is a virus able to infect a person shortly after it has just been cleared? Well, my first reaction to total casualties in Omicron in just an 8-week period worse than 23 weeks of Delta is that it infected a lot of people. Okay, mostly a population thing. The infection itself wasn't any worse, but it spread through the population really rapidly and infected a lot of people. I don't think the fraction of those people who were seriously ill was significantly different than in Delta, but there were a lot more of them. So you saw a big peak. Yeah. And why it is that it seemed to spread so fast, I don't know. But the fact is that it can escape some fraction of the pre-existing immune response. So that would capture both a lot of people who'd been previously infected and people who had not. So it has, potential to infect a lot of people. Any thoughts?
I think I would agree. Yeah.
Me too.
And I find it amusing that, remember, when Omicron first emerged, the press was calling it so mild you could be like a cold, right? And Daniel Griffin kept saying, no, I'm seeing people die of them. And now we realize that it is just as not mild as Delta was. Yeah.
Right. Possible mechanisms of COVID infection relapse. I don't think.
Is this the Paxlovid story? Is that what he's talking?
I don't know. Episode 903. Was I there? I don't know of COVID relapse. I know of reinfection. Well,
there is. No, that's Clinical Update with Daniel Griffin. Okay, so that's Paxlovid relapse. Yeah.
Okay.
So that's where this situation where 1 or 2% of people who get Paxlovid, after the 5-day course, they feel better, they become PCR negative, and then a few days later they have PCR positivity and they may feel bad. But that was also observed during the clinical trial. So this is nothing unique. And I think Daniel has said this is just in some patients for reasons that are not clear. the 5-day course is not sufficient to clear the virus. So it's not actually cleared. It's just reduced to a low level, and then it comes back for some reason. But then it clears again. So
Yeah, like maybe it was in sort of an immune privileged site and then
Yeah.
it comes out or something like that.
could be. Okay, Rachel writes, hello, and thank you for taking my question. The patience and endurance of the entire TWiV team is remarkable. We would be lost without you. In 2020, I heard a lot about viral dose explaining how sick different people get from COVID. Is that still a thing? Or is it a false hope that something explains illness severity, not predicted by age, health, risk factors, etc.? So I remember when this was being thrown about, I never bought it because there were no data, right? Nobody measures. I mean, first of all, we have no idea how much virus people inhale. And so hardly anyone measures infectious virus to begin with, so we don't know how much is shed. So there was just no way to make that conclusion. And I think now people realize that it's not a thing. No, it's a false hope that was spread by people who probably weren't familiar with what needs to be done. Now, the challenge study, which was done in the UK, they used just one dose. And you could imagine that maybe in a future challenge, they might address it. But, in that challenge study, I think they gave them 50 TCID 50s, and half of them,
Right.
and half of the people got some kind of COVID, pretty mild COVID, although one person did lose sense of smell. I guess you could try putting more virus, but I don't think that's, and I remember us talking about this on TWiV, I don't think the dose is going to necessarily control the illness severity, rather the kinetics, right? You may just go through faster. Anyway, I don't think there's any evidence for it. What do you guys think?
Well, I would just point out that I remember these discussions. In a laboratory situation, if you do a dose escalation study of a given pathogen in mice, you quite often observe that you get either quicker or more severe disease with higher doses. But that's a very specialized situation, okay? I think pretty often doses of pathogen that are outside of the physiological range, outside of what you would ordinarily, and delivered directly to a target site or artificially introduced to an abnormal target site, et cetera. So I don't know, there may be out there, but I don't know of any, generally, any such studies in humans with any given pathogen. There may be some, and certainly not with SARS-CoV-2. We don't know.
Yeah, it's sort of like you're pointing out unknowns on both sides, because as Vincent points out, we don't really know how much is sort of the physiologic amount you are potentially challenged with. And then as Rich points out, we don't know what the levels of variation are to actually look at for this.
Yeah.
We don't know what an infectious dose is.
Nope.
Though, as I recall, and I forget what the number is, but there has been a study that estimates the infectious dose for, it was either SARS or MERS, one of the two. And frankly, I forget what it was. It wasn't real high. It was, in the 10s or 20s.
Brianne, can you take the next one?
Sure. Reed writes, "Hi, TWiV. Hope you are doing well. I am a student from Caltech in BI 115, a course on viruses and their applications to biological systems. I had a quick question about the mice discussed in TWiV episode 900, Burning Down the Mouse. When you were discussing creating the immunodeficient mouse models for COVID-19 testing, you mentioned that they were injected with human fetal liver stem cells to, in theory, create human hematopoiesis in mice. I'm wondering, wouldn't it be more valid to introduce stem cells from the adult bone marrow? Do you think this would affect the results, considering most people who contract COVID have hematopoietic stem cell generation in the bone marrow, since bone marrow has a different microbiome and cells there have likely accumulated mutations? Best, Reed." I think that's a really interesting question, but I think that the idea with this model is that you're not necessarily just looking at the cells that were developed through hematopoiesis during infection, but you need to have a pre-existing pool of cells. like if you were a human, yeah, you would have some current hematopoiesis going, but you also have cells that were made in your fetal liver way back when you were a fetus. And so since the idea here is you're first populating the mice, it seems to make more sense to do it this way. I also think it's a lot easier to get fetal liver stem cells than adult bone marrow, given that the adult bone marrow is mostly going to go for transplant in humans as opposed to these types of mice.
Rich.
You want me to take the next one? Is that it?
Yes, please.
Ben writes, I'm a longtime supporter, listener of TWiV, and I can honestly say that your podcast kept me sane over the past 2+ years with the deep fact-based approach to understanding the SARS-CoV-2 pandemic. I'm writing today to ask a question that has hit close to home for me. 2 weeks ago, my wife experienced sudden hearing loss in her right ear. Most of the loss is low frequency, and her ENT has prescribed a course of oral corticosteroid, prednisone, as well as a series of steroid injections into the ear. She is right in the middle of the treatment and so far hasn't seen any improvement. What makes the story a bit more interesting, complicated, is that she is double vaccinated, Pfizer, and has been boosted once. While she has been careful to protect herself, she came down with COVID-19 in early April and suffered a mild case, fever for a few days, sore throat, cough. After making a full recovery in mid-April, she got what appeared to be pink eye, which self-resolved in a few days. In the middle of May, about a month after the COVID infection, she experienced the hearing loss. From our investigation and speaking with the ENT doc, hearing loss is highly complex and can be caused by a number of things. One notable cause is viral and related to inflammation. It is tempting to consider if her recent COVID-19 infection could be related to the hearing problem. If it's a potential cause, would it be worth trying an antiviral, since we're throwing the kitchen sink at this problem, to see if it would help? I deeply appreciate any insights from the TWiV team. Kind regards, Ben. Well, this would certainly be the first example I've heard of a hearing loss associated with a SARS-CoV-2 infection. So, based on what I know of SARS-CoV-2 pathogenesis, it seems unlikely. It's also, the history is somewhat complicated by the pink eye. We're talking about pink eye can be caused by bacteria or viruses, but there's an adenoviruses are associated with pink eye. So that's she's got another infection going on there.
I see the history part in terms of the infection in early April and the hearing loss in mid-May being maybe less direct viral and more about post-viral inflammation.
Yeah.
But I actually have no further evidence on that, and I can't say.
So, Brianne, you make a good point. From that point of view, an antiviral at this point is kind of way too late.
Right.
If there was even a viral etiology.
That's also I don't know, what does Daniel think?
Yes.
Well, so he's interested in whether Paxlovid would help long COVID, right? Mm-hmm. Is there any evidence that there's some virus around for a long period of time that's doing that? So if that trial isn't done by others, he may do it himself. I don't know, but I think it's too late, right? The virus is long gone, but
I suppose that if this were this is compound ifs, and compound ifs are not really permitted. But if this were a long COVID thing, and if hearing loss is part of long COVID, and if long COVID in part represents a lingering virus infection, then
it might be,
it might make sense. But that's a lot of ifs.
But hearing loss is very rare. What is not rare is tinnitus, right? The ringing happens after COVID. Remember, we've heard that from the beginning. And so there's something going on in the ear. It may be inflammation. And so I just not sure an antiviral would work, but you can't just use it because it's the EUA for Paxlovid is not for hearing loss, right?
Right.
All right, one more. Philip writes, I find the weekly TWiV podcast to be informational for COVID-19. I appreciate the monkeypox updates as well. I've watched many of the older TWiV podcasts and find the episodes on polio, smallpox, and rabies to be fascinating. I only heard one reference to coronaviruses pre-COVID. Actually, we did a number of corona. We had Matt Freeman on, and also the guy who captured the bats. What was his name? Donaldson, Eric Donaldson.
Yeah.
At least 2, and probably more.
Yeah, but that's out of what we were at episode 535 when COVID struck. So if there's only a couple of pre-pandemic COVID things, you're looking for a needle in a haystack.
Rabies is an example of another disease that can be mitigated with vaccines. It will always be in the wild, but every case in domesticated animals is preventable, as well as human cases from those animals. My insurance covers pre-exposure rabies, so I took advantage of that. I do plan to spend a lot of time in Colombia in retirement, the country, not the university. The full post-exposure treatment for those without pre-exposure vaccination is expensive and may be hard to find outside the U.S. Yes, you can get, certainly certain people who work with animals get pre-exposure rabies vaccine, right? Veterinarians, people who are out in the woods doing whatever. But if you're going to be in that risk place, you can get, if you're going to a country where there's a lot of rabies, you could get it as well. Sure. All right, let's move on to some picks. Brianne, what do you have for us?
So I am unfortunately not going to ASM Microbe, but one of my favorite parts of ASM Microbe each year is the Agar Art contest. And I was trying to read about the current Agar Art contest and found out that they actually have a microbes rule exhibit at the Liberty Science Center, which is pretty close to here, that shows some of the Agar Art. And so it's an entire microbiology themed exhibit at the Liberty Science Center. And I am now making plans to get to the Liberty Science Center, since it's very close by, to go check this out.
Excellent.
Oh, that's neat.
That's cool.
You know, one year someone did an Agar Art of me.
Yes, I remember that.
But it didn't win. I voted for it because I was on the the committee that was what do you call it? Judging them, right? And I guess it was not quite right that I voted for myself. I'm trying to find it here. But I just doing a search for Agar Art Racaniello doesn't pick it up.
So
there it is. There it is. Yeah, it's TWiV in the modern world. Yeah, so someone wrote a nice little spiel about it. It looks pretty good. Yeah, my glasses are 2 different colors, 2 different back there. Look at my beard. Have you guys seen this?
Yeah, I remember it.
Yeah.
It looks really good. I'm sorry to talk about myself.
Brianne, the Liberty Science Center, what is it?
So it is this place in sort of near Newark. Basically, the Statue of Liberty is officially in New York, but Liberty State Park is in New Jersey. And at that area, there is this science center where there's sort of some exhibits. We have a lot of students go there and do presentations. It has a bunch of different science-related exhibits and other activities.
Cool. They've got a thing called Minecraft to the exhibition.
Yeah.
My grandson would love that.
Yeah. And so I have, I see the signs when driving in that area pretty often. And now this makes me think, why have I not gone to see Microbes Rule? Because Microbes Rule.
Yeah, cool.
I've been there a few times. I was there on I think it was a Boy Scout or Cub Scout trip. I don't remember which. And we slept in the Bloody Museum overnight. I don't know why they thought that was a cool thing to do, but there's this big room where all these kids put their sleeping bags, no tents, just sleeping bags. You got 50 kids and there's a lot of noise. You think I got a wink of sleep?
That's that's sort of a horror film plot, I think. Night at the museum.
I mean, the idea would be you wake up and you go check out the museum. But I just remember there was a HIV exhibit, and they had so many things wrong in it scientifically. It really pissed me off. And then I look and I see a number of people I know who are on the advisory board. They didn't pick any of it up.
Well, I will check this out and I will report back. And Rich, you can then bring your grandson.
Excellent.
It is a big place, though. Yeah, it's on the way. If you drive to the Holland Tunnel, it's right there on the way to the Holland Tunnel. You can see and it has a very nice planetarium, right? Where, you know, you can sit and you got the big dome and stuff. We went in there as well. That's pretty cool. Rich, what do you have for us?
I just wanted to impart a brief Artemis 1 update. This is Artemis. The Artemis program at NASA is a program that has the long-term goal of sending people back to the moon. And I think I'd have to confirm this, but I think it even establishes a longer-term presence on the moon. But Artemis 1, the first phase of this project, is to send an unmanned mission to orbit the moon and bring the capsule back. And that has the effect of basically testing all the technology. So I got 4 links here, one to the NASA Artemis 1 site to start with. Another that I found poking around is a mission diagram. It's a PDF from NASA that shows the steps of the actual flight from takeoff to an Earth orbit, translunar injection, a bunch of fiddling that goes on while you're traveling to the moon, and then a weird orbit around the moon, and then coming back to the U.S. And then 2 links to articles in space.com, one of which is a nice sort of slideshow of photographs from what they are anticipating at different, this was published back in January, different stages of the mission, with a few little extras about how they're going to have a mannequin on board to measure what's going on, and a couple of little gravity indicators, one of which is a Snoopy in a spacesuit, and they're taking a couple of Lego spacemen with them as well. And then lastly, this is what I was looking for to start with, was launch dates, possible launch dates for the status of the program. Now, this is going to go up in the new Space Launch System, which is a giant rocket with a couple of solid rocket boosters attached to it, and the capsule itself is a newish capsule called the Orion capsule. And where they are now is they've been doing, I think they call them wet tests, where they roll this thing out to the launch platform and fuel it and get it right up to what they would do to launch it, to make sure all those systems are okay. And then they dump out the fuel and roll it back and prepare for the actual launch. Well, one or more of those wet tests didn't go well. There was a stuck valve or something like that, and that has delayed the launch. It was supposed to have gone off already. So they're rolling up to the next wet test, and they have launch dates all the way through the rest of the year. I think the prime target is somewhere late in August. So I'm looking forward to that.
So is this basically similar to the other moon launches? They send up a ship that breaks into 2 and one lands, and then...
Well, for this, they're just going to orbit the capsule.
Okay.
I would have to look at the whole Artemis program again, because and see what the ultimate structure of this is, whether you have an orbiting bit and a landing bit.
Yeah.
I think it does, but I would have to review that. I will do that and come back to you with a future pick.
In the old original landing, there was a lunar excursion module that separated from the capsule, right? And that went down, and then only part of it came back up again. They left something behind, and then that docked, and then they crawled through and they left it, right, and all that stuff.
Yep. Yep. Yeah, and by the way, Kate Rubins is on the Artemis team.
Really?
Oh, yeah. So theoretically, she's a candidate for for being the first woman on the moon.
Very nice.
She's a candidate for that. Okay. So maybe we'll get to do a TWiV with Kate from the moon. How would that be?
That would be cool.
I vote yes.
So she was originally going to be at Microbe, and we were going to try and get her on TWiV, but then she canceled. Maybe she has to do Artemis stuff.
Who knows? I'm sure
That would
she's
be very
plenty
cool.
busy.
So last week, Brienne had a 3D printed ear. So that reminded me of this video that I watched, which is about a 3D printed house. There's an Italian company called Tekla, and they built, I forgot, it's an acronym for something. They built a machine to 3D print houses, and basically they make a mixture of water and something that solidifies, and they have these big arms that 3D print it. And they make these little kind of roundish homes. They put two of them together and can have a kitchen and a bedroom or something. And they're pretty cool. The technology is really neat. And this particular video is talking with the architects and the developers of this, and they're Italians, and they're very enthusiastic about this. The video is called Eco-Sustainable 3D Printed House. I don't know if it's eco-sustainable because, once you print the house, you still have to put windows and doors and a roof on it. And it takes, I think the 3D printing is a minor cost of the whole house, but it's a very cool technology. And like everything else, it's going to evolve. And it's really neat to see how they, the arm, it's squirting out this clay material, and it does it in circles to form the walls. It's a pretty thick wall, which is made up of multiple circles, and it goes around and around and builds it up. And then the outside has the shape of that. You can imagine stuff being put on it. Anyway, it's very cool to look at.
I like that they're making this out of earth.
Earth, basically, yeah.
Yeah.
Yeah. So in that way, it's definitely at least very eco-sustainable.
So there's another video on YouTube you can find where they basically say this is not ready for prime time for a number of reasons, and can't make it. I mean, we need, apparently there's so many homeless people. How many did I hear last night? Like 100 million homeless people on the planet. That's going to go up to 1 billion in 7 years. We need 96,000 new homes a day to accommodate them. And I don't think they can print that many, but maybe they have to scale it. Anyway, it's a very cool technology. So,
well, this is the kind of thing that sort of tweaks my optimism about the species, because, I probably spent too much time watching the news, and it can be, you can come away with a pretty pessimistic
for sure.
Feeling about our future. But humans are pretty clever.
They are.
And unfortunately, they don't really get their act together, especially politically, until there's a crisis. But in the background, there are a number of imaginative people anticipating these crises and coming up with really interesting solutions. And this is one of them. And it makes me feel good to see that people are working on this kind of stuff. It's great.
And one can only hope that the imaginative people, the ones that get us to the moon and that do this sort of thing, can prevail, right? Because, we're on the verge of being an autocracy, right?
Well, I think my impression is that what happens is that when a crisis really arises and things are really collapsing beyond the control of the politicians to keep it together, and everything is collapsing around them, then these clever people rise to the surface because they've got the solutions, okay, and they can implement it.
There are a number of countries that have gone the autocratic route, and it didn't seem to have worked there. So hopefully we'll do it differently.
Yes. Speaking of news, Rich, I just got an alert that the vaccine advisory panel has voted in favor of Novavax.
Yes.
And it may be distributed by the end of the week.
Cool.
Yep.
Very good.
Cool.
That's good. And Paul Offit's going to come back on TWiV in a couple of weeks because I asked him what he thought about another booster, and he wrote, I think we've lost our way. So I said, why don't you come on and explain it? And he has some advisory panel. He says, I can't come on before that. So he'll come on after that. That is TWiV 907, where we are optimistic and want things to be good. We don't want them to be autocratic. You can find the show notes at microbe.tv/TWiV. You can send us your questions, comments, picks of the week to twiv@microbe.tv. If you enjoy what we do, you could support us. Well, what is it that we do? We make science content, and it's long, but we try and get experts in various fields to talk about what they know. So if you like that, you can go over to microbe.tv/contribute and make a donation, and it will be U.S. federal tax deductible. Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
And Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.